Thank you very much for inviting me. I've been to Ireland only once before, and that was for a very short time. And as I was saying to some people earlier, we were put up in a very, very fancy hotel. It was in a castle, and we were very uncomfortable because it was so fancy that we moved out and stayed in a little B&B. So this is wonderful. I'm really looking forward to walking more in Galway and seeing more in the city. Um, when I was first invited, I thought, oh, I'll tell you about the past and the future. And the more I thought about it, the more I realized I don't know what's going to happen in the future. As you just heard, it's very hard to predict the future. Um, and then I started reading what you at TMRN are doing and realized and I was very, very impressed by your outreach to children and adults, teaching them what clinical trials are about, what randomization is, what experiment is. And it occurred to me that while we have a lot of advances, oh, I realize I should be watching my watch, a lot of advances in clinical trials, I think we don't do as a community as much outreach to the public as we should. So that was what I was going to talk about. And I will talk about that a little bit. But then we had an election, a totally disastrous election that is going to mean, I think, not only should you in Ireland take leadership, you're going to have to. We in the US are going to try to hold the fort, try to keep people involved, but it's going to be a terrible 4 years. So we need you, we need you, we need England, we need everybody else beside the US to take over, to take leadership. So, oh, and there's some disclosures. So I'm going to talk about, the theme will be, this is sort of, I'm going to turn this around so I can see the slide, yeah. That the success or failure of the trial depends on the participants themselves. Those of us who work in the field, it's important that we do a good job. But if the participants aren't involved, if they don't understand what they're doing and why, a trial can fail. Now, I want to make sure that I'm clear. When I say a trial is successful, that doesn't mean it's shown the benefit of treatment. It means that the data are interpretable, the results, you can understand what they're saying. That, to me, is a successful trial. And a failed trial is a trial not that hasn't shown benefit, but that you can't understand the data. The data are uninterpretable. So that's what I want to stress. And as I have been reading what you're doing, I have been very, very impressed by your training of young children and the training of adults. And so I want to give you a history, a personal history of what I learned as a child about randomized trials, and then 40 years later, what I learned as an adult. In both cases, I'm talking not about how the trial is run, but really what the participants in the trial think about the trial and think about what they're doing. So in 1950, my family and I moved from Brooklyn, New York, to New Milford, Connecticut. Brooklyn is then a city of 3 million people. It's one of the boroughs of New York, but it's the biggest borough. So it's a big urban place. And we moved to New Milford, Connecticut, which is a tiny, well, maybe compared to some of your villages, it's not so tiny, but compared to Brooklyn, it was tiny. It was 6,000 people. It had a village green, and around the green, there was the library and the elementary school and the Congregational church with the white steeple and the high school, and then at the top was the hospital, and the bottom was the city hall. So everything was around the green, and there was as you can see here, this is actually a picture of that green. There was a gazebo, and that's where the band came and played. So it was a very small, lovely little community. And then this is a picture of second graders. It's not a picture of my second grade, but it's what second graders looked like in 1950. And in the summer, we would go swimming in Candlewood Lake, and there's a picture of Candlewood Lake in New Milford. That's the largest lake, the largest man-made lake in Connecticut. And so when school was out, in second grade, we went swimming in Candlewood. That was in the end of June and the beginning of July. And then polio came. And when polio came, we were not allowed to swim anymore because that was where kids caught polio. So this is then the summer of 1951. I had just finished second grade. And then in early August, we moved to Danbury, which was 17 miles away, and it was 30,000 people instead of 6. So it was a reasonably large town. My mother kept in touch with the parents of that second grade, and 3 of the children, second grade was about 25 kids, 3 of the children ended up in an iron lung. They all lived, but they were there for 3 weeks to 3 months. And my next door neighbor, who was a little 3-year-old kid, died of polio. So it was a lesson in, it was our version of COVID. It was a 7-year-old's version of COVID. And then in 1954, so now I'm in 5th grade, but my brother's in 2nd grade, we're in Danbury. Second graders, there was a polio vaccine trial. It was very interesting that there were 2 parts of the trial. There was a randomized part, randomized 2-to-1 to get the vaccine or placebo, 600,000 kids. There were only 11 states that allowed randomization. Those states, interestingly enough, are pretty much the same states who voted against Trump this time. So 50 years later, it's kind of the same. And the results were very successful. That the 60 to 90% protection against polio vaccine, the virus, and 94% effective against the development of bulbar polio, which is what led to the iron lung. So my lesson as a child, my brother was one of the people, he was in 2nd grade, so he got the vaccine. Oh, it was good to do experiments, but there was resistance to randomization. And so, as I say, 11 states randomized, and that was led by New York. The Public Health Department of New York, Francis insisted you had to randomize. 33 states refused to randomize. And then there were 4 other states that didn't participate at all. Connecticut was one that refused to randomize. So my brother, we knew, got the polio vaccine. And so in those cases, there were 1.8 million children who got vaccinated in the non-randomized part. You could volunteer, or your mother would volunteer you, maybe your father, but in those days it was almost always the mother. The second graders got the vaccine if they volunteered, and the first and third graders didn't. Those were the controls. And of course, it was very hard to interpret those data because you didn't know who would volunteer, and that's a self-selected, and then you didn't know whether second graders behaved differently from first graders and third graders. As a child, I was going to call my brother and say, Johnny, do you remember getting vaccinated? I remember that he did, but I was old. I was in 5th grade. But what I wondered as I reviewed this and thought about my own memory, would we have known more? Would training of people about randomization have prevented this wasteful 1.8 million vaccines? Okay, but I think it would be interesting. It's probably too late to know because those of us who are, those who were second graders then are pretty old by now, how much of that experience taught them, and did that experience last for life, about the importance of being part of a trial. For me, as a child, seeing my brother and being jealous that he was able to get the vaccine and I couldn't, it was actually a very influential trial, personally. And when I see what you people are doing for children, I think it's really, probably really important, that it will encourage them to participate in trials. And then I'm going to tell you a little about my experience 40 years later. I chaired the data monitoring committee for the Women's Health Initiative, which looked at, among other things, the effect of what was then called hormone replacement therapy, it's now just called hormone therapy, on postmenopausal women. And let's see if I have, well, yeah, no, sorry. So what happened was, so now go back to 1987, something like that. The data from epidemiologic studies had shown a 50% reduction, well, I shouldn't say reduction, that those women who were on hormone replacement had only half as much coronary disease as measured by MIs as those who were not. And so the question was it the hormone replacement therapy that did that? And most people believed it. So to do the trial was a huge battle. But the question was, if you gave women hormone replacement therapy, did it prevent coronary disease? So in the data monitoring, and we all believed that, we at the data monitoring committee believed that, well, of course it worked, but it was important to do a trial because we believed in trials, but we knew what the answer was. So it was surprising when the data started coming in that what Salim Yusuf said, he was on the committee, he called the 3 meanings. I don't know, do you guys use the word meaning? Bad things? I'm not sure whether that's an Americanism or not. Heart attacks, strokes, and pulmonary emboli. Those all increased in the women who were randomized to hormone therapy. And it was very scary as the DSMB, because, and we perused the informed consent document really carefully. Did it warn that? And the answer was no. It did say something that you could interpret as maybe it increased the risk of stroke, but certainly it didn't give a hint that there could be something bad doing to your heart. So we struggled a lot and finally said to the sponsor, which is the NIH, the NHLBI, National Heart, Lung, and Blood Institute, you have to tell, the investigators have to tell the participants that we're seeing increases in heart attack, stroke, and pulmonary emboli. The investigators didn't like that recommendation. The Heart Institute didn't like that recommendation. They said, everybody's going to quit. And we said, well, sorry, guys, we can't not tell them. We have an informed consent document that told them one thing. We're seeing data that's saying something else. You have to tell them. So they did. And mirabile dictu, hardly anybody dropped out. Later on, after the trial was over, and we eventually stopped it for harm, by the way, but when it was over, people went to the individual. There was a large study of why did these women stay on, and the answer that they got from many of the women was, we're here for our granddaughters, not for us. And that to me said, wow, we need people don't join trials necessarily only for themselves. There's an altruistic piece of it. And so then I went, just, before this talk, I went and I looked at informed consent documents. Do they talk about altruism? And the answer is a little bit. They talk about the importance of science and so forth, but they don't come out. Mostly it's, what does it do for you? What's the benefits? What's the harm? But very little about what it's doing to society and what you as a participant are contributing in an altruistic way. So it seems to me that one of the things we ought to be doing, in addition to everything you're doing, is check, and it's always hard to generalize from a sample of size 1. Maybe other trials don't do this, but make sure that the informed consent documents and the investigators stress the altruistic nature of joining a trial. So then what I had planned to say before we had our horrible election was we're in the cusp of great advances, that we have really good methods for clinical trials. Biology is amazing. We have really good methods for, you described earlier how you're increasing the ability to recruit. So the operations of trials have become very, very mature, and well-done trials are done really well. The computers, the databases are becoming more and more sophisticated. The statistical methods, which is where I come from, are more and more tailored in ways that are very positive. And so we're in a state where the future looks bright. And then I wanted to show you some examples of these amazing, to me, amazing results. These are 4 trials, all of which I've been involved in. And what's striking about them all is how big the effects are. So the first one, right here. This is a heart failure trial from dapagliflozin. The primary, so these are people with class 2 and 3 heart failure, so they're sick, but not very sick. The endpoint is an important endpoint. Heart failure hospitalization or cardiovascular death. And the hazard ratio is 0.74, basically a 25% reduction in these important endpoints. The important point here is that the upper end of the 95% confidence interval is 0.85, far away from 1. A p-value is a little tiny p-value. This is a dramatic benefit in people with pretty serious heart disease. Here's cancer. This is, I can never pronounce the name, so I just call it IPI. It's metastatic or recurrent non-small cell lung cancer, so a bad cancer. The endpoint is overall survival, so that's a pretty strong, unambiguous endpoint. Hazard ratio, 0.69, so a 30% reduction in overall, and 30% improvement in survival. And again, the upper end of the confidence interval is 0.87, far away from 1. This is a really dramatic result. Hazard p-value 0.006. So these are 2 major diseases with great big effects. But these are large sample sizes, of course, because these are the major diseases. But here are orphan diseases. This one is amyloidosis. It's a specific kind of hereditary one, where the endpoint is it's a quality of life endpoint, where the patients are followed from baseline to 9 months. They get worse quickly. Whoops. And the placebo group, these were randomized, there were 76 placebo and 121 active. The placebo group gets worse over these 9 months. The treated group gets better, a dramatic difference in not a very large sample size. And here's gene therapy this is a very specific retinal disease, disease that inexorably leads to blindness. And the design here was, so here in the amyloidosis one, increases is bad. How can this moment move? In the SPARK trial, increase is good. So as you over time, if you increase, that means you're seeing better. So the randomization, there were 9 placebo patients and 20 active patients. So this is a tiny sample size. The placebo people's vision stayed essentially the same for the first 9 months, and the gene therapy went into the worse eye, the eye that whose vision was worse. The active patients, had an immediate benefit that stayed pretty stable. At the end of the first year, the placebo patients, the other eye got treated, and the other eye, which had been the better eye, of course, improved. So here we're talking about a dramatic result, and I think when we see gene therapies, we're going to see more and more dramatic results with tiny sample size. So that's the positive. But there are threats. And I want to mention 3. You may think of more, but these are the 3 that worry me the most. First is scientific threats, people who don't believe in randomization and argue against it. Then there's the internal threat. For those who do argue for randomization, but who argue against, they don't say it that way, against rigorous analysis. And then there's the external, the US election, which I'm going to say a few more words about. So there's the scientific threat, the drive to replace randomization. The argument is trials are too expensive, they're too long, they take a lot of time, sometimes the results come out and they're out of date, participants aren't a representative sample of population, et cetera, et cetera, et cetera. There's a whole lot of criticisms that are made. And we have magic potions. We can use historical controls. We can use digital twins. We can use non-randomized concurrent controls, like first and third graders back in the vaccine polio trial. There's artificial intelligence that can figure everything out, and so forth and so on. There's a very strong defense. If you haven't read it, the New England Journal, Magic Randomization versus the Myth of Real-World Evidence from the Oxford crew. And I do think there are times we can believe in non-randomized studies. And the dirty little secret is very often non-randomized studies show the same results as randomized studies. The problem is we don't know in a particular case whether it would have or not. In a non-randomized study, the biology has to be compelling. In a randomized study, it doesn't. It's nice if it's compelling, but if you can show that something works and you don't understand the biology, it probably does work. The control in a non-randomized study has got to be concurrent or nearly so, or you have to have a convincing case that the disease hasn't changed over time and that the other therapies haven't changed. By definition, in a randomized trial, you've got concurrent controls. The outcome has to be objective. You don't need objectivity. It's good to have in a randomized trial, but you don't really need it if you do the trial in a way that protects you from a non-objective outcome. And in a non-randomized study, the effect has got to be large. It's got to be so large that you say it couldn't have happened by chance. And it's not needed in a randomized trial if your study is large enough to identify the effect. So in general, for a non-randomized trial, you've got to have stronger evidence than you do in a randomized trial, or I would say stronger apparent evidence because of the lack of rigor that a non-randomized trial has by definition. Okay, and if you, the JAMA, the Journal of the American Medical Association, has a reasoned standard, and I think if you haven't seen it, you should look at it. This is a way of saying there are cases when non-randomized trials are fine. But when we need randomization, and I say we almost always do, randomization is necessary but not sufficient. We need data on all randomized participants, and that's why it's so important for participants, not only the investigators, but participants to understand once you're in the trial, you better be there at the end. We must not use post-randomization data to distort the benefit of randomization. But, and this is the thing that I really want you all to fight, the International Conference on Harmonization, E9, which is the statistical principles for clinical trials, R1, which is the revision, the revised version, it does exactly that. It actually promulgates the use of post-randomization events that distort the balance conferred by randomization. Regulatory guidances should move us forward. My contention is that R1 moved us backwards. It deals with intercurrent events like death, like going on to another therapy, like crossing over, all kinds of things that happen. And they say, okay, so R1 says, okay, you do that. We have a way of dealing with them. There's the hypothetical product. Oh, this person died, just pretend they didn't die. And assign data to that person as if the person didn't die. Oh, they changed treatment, pretend they didn't, just pretend they stayed on. So that to me is, you can't pretend things that didn't happen. You know it, you know the person died, you can't pretend the person didn't die. There's on-treatment analysis, or its partner while alive. Ignore all the data after the person stopped treatment. Well, that's part of the treatment. Part of the treatment is maybe not finishing. Can't do that. And then there's principal stratum analysis that says, they had an IC, an intercurrent event. Just don't even include that at all in the study. The problem with this is that it's now the regulatory standard. And so I think we have to fight it. And it's going to be hard to fight. But, and this is a quote from David Sackett, who was the Canadian who, actually he was born in the US, but he's really Canadian. This is from a chapter, an unfinished manuscript that he was writing as he was on his deathbed. And he sent me his first chapter, which said, per protocol analyses, which is basically what R1 is promulgating, confined to compliant patients, this is the principal stratum, are inherently invalid, and I consider them nefarious when carried out by folks who know better. And now there's the external threat, and that's the US election. I wasn't planning on talking about this because I was planning for Trump to lose, but he didn't lose. And this is our new likely head of Health and Human Services, Bobby Kennedy Jr. For those of you who don't know how the US is structured, or this part of the US Health and Human Services, in addition to having Social Security and all kinds of other things, it has 3 agencies that are really important to us in clinical trials. It has the National Institutes of Health, that does medical research, funds it does it internally, and it funds it externally. The FDA, the Food and Drug Administration, which regulates drugs, biologics, medical medical devices, and veterinary medicine, and food, but I'm talking on the drug side, the drug and medical devices. And the Center for Disease Control, which its role is the protection of public health. So for example, if you think of vaccines, COVID vaccine, for example, or flu vaccine, that gets developed, at least part of it, at the NIH. It gets approved or by the FDA, and then the promulgation of who should get the vaccine, that goes to the CDC. So let me give you an example of some of the things, and Kennedy will be in charge of all of this, unless the Senate doesn't approve of him, but I don't think that's going to happen. So I'm going to give you 11 things he said. AIDS may be caused by poppers rather than HIV. And poppers was an inhaled drug popularized by gay men in the 70s. 2, the 2020, and these are only 11, there are many more. These are the ones that I didn't have time for more. The 2020 pandemic was a plandemic, an event orchestrated by the government as part of an effort to undermine American liberties. Number 3, COVID-19 is targeted to attack Caucasians and Black people. The people who are most immune are Ashkenazi Jews and Chinese. Whoop. Whoop. He favors raw milk, ivermectin, which is for horses, and hydroxychloroquine. He doesn't want fluoridation. He wants to see medical journals, he wants to sue medical journals for being patsies, and that's his word, to the drug industry. NIH is little more than an incubator for the drug industry, and he wants to, in the first day, get rid of 600 people from the NIH, and he wants those people to be the influential people. Vaccines have led to autism, and we know there was a study that came out in Science years ago, and it's been repudiated over and over. Vaccine scientists should be put in jail. This is a direct quote, put in jail, and the key should be thrown away. And number 11, they're putting, this one's really crazy, they're putting in 5G to harvest our data and control our behavior, digital currency that will allow them to punish us from a distance and cut off our food supply. This is the person who's going to be head of NIH, FDA, and CDC. There's a glimmer of hope, which was on a couple days ago, and I was hoping that by today I could get rid of these slides, but I don't think so. So placing Mr. Kennedy in charge of DHHS would put the public's health in jeopardy and undermine America's global leadership in the health science, and that's the message to you. What can you do? Our leadership is going to go away. Even if Kennedy isn't chosen, there's no guarantee, there is a guarantee that whoever will be there will not be good. You and other nations need to, it's not just because it would be good to, you need to take leadership. We're in limbo. Maybe it won't be as bad as I fear. I've spoken to a lot of people at NIH who are leaving, people who've been there, FDA, who are leaving. They've been there for a long time. They're going. They don't want to be fired, and they don't want to be in an administration like this. So we're passing the gauntlet to you. You and the rest of Europe and the rest of the clinical trials community, you've got to take over. We can help, but we can't lead. So what should you do? And you doesn't equal us. It doesn't equal the US. Hold tight to lessons of the past. We've learned a lot. We know how to do good trials. We know how to ask good questions. Listen to critics. If somebody argues for non-randomization and it makes sense, that's okay. Remember the lessons that I described from polio and WHI. You may each have your own lessons, that participants view their roles at least partly altruistic. and continue what you're doing, teaching the public, young and old. Fight violations of randomization, and that in particular, fight R1. I have a paper that co-authored a paper that got totally rejected because it fought R1. We now have another paper to another journal. We're hoping it'll get in, but it's very hard to fight authority. And as I say, you've got to seize the opportunity of leadership. We, the U.S., will not be able to lead. We're not going to be able to come back at least for 4 years, but we don't know what will happen after that. And the landscape will have changed. So I toss the responsibility to you. You've got the expertise, you've got the personnel, you've got the conviction. Do a great job, and we'll be back in 4 years.